So in the second example in Lab 4, we are going to be using orthogonal contrast again, but this time to do a trend analysis. And this is what the second script is going to walk through. Here we have an experiment in which we have plants at different spacings and we're looking at yield. And so we have 5 different plant spacings in the field. And for a trend analysis, we are not interested in comparing the yield at different spacings per se. We're interested in the overall relationship between plant spacing and yield. And so this is why we're doing a trend analysis. If you pull up the script for this, so here's example 2 script, and let's pull in the data set and take a look at that. You'll notice in the script I refer to the data set as spacing.dat. So I'm going to replace the name here. Spacing.dat has a header. And you see here we have replications or plots. We have 6 reps per spacing level. These spacings are the plant spacings are evenly spaced, 18, 24, 30, 36. And it's because of this even spacing between our treatment levels that we can use orthogonal contrast to do a trend analysis, and then we have yields with all of those. I'm not going to spend a whole lot of time in the initial part of the script. The first part of the script, we're going through checking the structure of the data set, making sure that the spacing is understood to be a factor, performing an ANOVA, and then partitioning those treatment sums of squares using our 4 degrees of freedom. Remember, because we have 5 treatment levels, we have 4 degrees of freedom. So in a trend analysis, we can ask for linear, quadratic, cubic, and quartic components to that response. The reason I'm not going to spend a lot of time here is because the template is exactly the same as we saw before. You're going to bind together the appropriate coefficients. And again, for these trend analyses, the coefficients are already given to you, the table I gave you in lecture. So it's just a matter of binding those together into the contrast matrix, assigning that to the appropriate classification variable in your model, and then running the analysis and pulling out the summary. And you see all of that. I go into that into detail in the lab handout itself. And we get a certain amount of results here where the 4 degrees of freedom are split into the 4 questions. We see a highly significant linear component, a significant quadratic component, but non-significant cubic and quartic components. And so in this particular case, you would interpret that there is a quadratic relationship between row spacing and yield. Again, I'm not going to spend much time on this because this is exactly what you saw in the first example. What I do want to do in this example is to do the same trend analysis using a regression approach. Now, to do that, let's take a look at our data set, structure spacing.dat. Right now, when we loaded the data set in, it was interpreting spacing as an integer because it's 18 inches, 24 inches, 30 inches, and so on. If we were to run this as an analysis of variance, spacing would have to be reclassified as a factor, and that's what you see early in the script here. We reclassified the spacing variable as a factor. For a regression analysis, it is not going to be a factor. It needs to be a numeric classification variable. And so if you come down here into line 43, here starting in line 37, I'm carrying out the same trend analysis that we did above using orthogonal contrast. And now we're going to use a multiple regression approach. And the first thing you have to do is reassign spacing as a numeric classification variable, or a numeric factor or variable. Doing that, we see that spacing is no longer understood to be an integer, but is now numeric. That's great. I should add as a word of caution here, if you bring in a data set, classify spacing as a factor, and then try to reclassify it as numeric after that, you will get some very strange behavior in R. So if you want to do some analysis with spacing as a factor first, that's great. When you go to do look at the regression approach, you will actually need to read in the data again, and that's what this line here is. Read in and inspect the data again. You sort of reset it as an integer and then reclassify as numeric at that point. a numeric regression variable. Once it is classified as such, and let's take a quick look at our data set, spacing.dat. We have 3 columns in the data set. We have replication, the spacing that it's at, and the yield. What you'll notice here is that now I'm going to define 4 variables, and those 4 variables correspond to the 4 potential regression components in a trend analysis. We have the linear component, which is just going to be spacing all by itself. So I'm taking the column of spacing and assigning it a new name. Let's just do that real quick. Now if I want to look at what that is, spacing, you see it's just a vector of numbers, and it's a vector of essentially the treatment levels. I'm now going to define 3 other variables, spacing 2, 3, and 4, which are going to be this original vector squared, raised to the 3rd, and raised to the 4th. So let's do all those. Take a look at spacing, let's take a look at spacing 2 down here. And you'll see it's a vector of the same length, but 18 has been squared to 324, 42 has been squared up to 1764, and so on. With these 4 variables in place, you can now write a very simple model. You can ask for an ANOVA of a linear model. And here you see I've taken the LM function. Usually I take that LM function and assign it an object name and then run an ANOVA on that object. And here I'm combining it all into one line. I'm running the linear model. That's what's happening in the highlighted area there. And then I'm running the ANOVA function on that linear model, all in one line. And the linear model in this case is that yield is explained by these 4 regression variables: spacing, spacing 2, spacing 3, and spacing 4. If you run that, you will obtain p-values associated with those 4 variables, each one of which has 1 degree of freedom. And you will find that when you do that, if you go back and look, if you were to do this analysis with orthogonal contrast, as we did in example 1, you obtain the exact same p-values for those 4 components that you got using orthogonal contrast, assigning coefficients and taking that approach. You can do it either way. You will get the same results either way. The nice thing about the regression approach is that you do not have the constraint of equally spaced treatment levels, whereas in order to use the table of orthogonal contrast I gave you, that table assumes equal spacing among your treatment levels. To that extent, the regression approach is more general and should always work. I have one final line of code in this particular script if you're interested. If you did want to find the best fit line, in other words, what we're saying here when we look at these results is that we have a significant linear component to the response and a significant quadratic component to the response. So that a best fit line, our model would contain a linear coefficient and a quadratic coefficient, and these non-significant p-values for the cubic and quartic components are telling us that the coefficients in front of the cubic and quartic term in the polynomial are not significantly different from 0, so they drop out of the model. So if we were to create a best fit line, we would need an intercept, we would need a coefficient for the linear component and a coefficient for the quadratic component. To get those, it's fairly easy. You just run the linear model again, in which yield is explained by a polynomial, and this is this R function poly. It's explained by a polynomial in which spacing is the regression variable, and it has 2 levels. And the reason we say 2 here is based on the results we got when we ran the analysis above. We only had the linear and quadratic component being significant. So we only need a polynomial of order 2 in order to for the best fit line for this particular data set. So spacing is the relevant regression variable. It's an order 2, and this raw equals true is what you would use in order to get estimates of what those coefficients actually are. Your other option here is raw equals false, in which you would be given orthogonal polynomials, but translating those into what the actual coefficients are for the best fit line is a little less straightforward. So spacing 2, raw equals true, and it's the same data set, spacing.dat. And if we run this, what you will see is that the intercept for your equation is 52.03. This negative 1.26 is your coefficient in front of the linear term, and here's your coefficient in front of the quadratic term. So if you were to pull all that together, and this is what I have in the lab handout here, if you were to pull all that together, yield would be explained by this particular best fit equation, where I've pulled those coefficients from the output into that one equation. Okay, that was pretty quick, but just wanted to show you how to partition again. Again, you're partitioning the treatment sums of squares into t minus 1 components, each costing you one degree of freedom. Partitioning the sums of squares and then assigning significance to each one of those components, which then allows you to formulate what the response really is between your treatment and the variable that you are measuring. That's it for this, but take a look at the whole script. Make sure you're comfortable with the orthogonal contrast approach for the spacing example here, and we'll pick up in the next video with multiple means comparisons.